Here at the University of Westminster, we're working on breast cancer, and we're particularly interested in understanding how the different subtypes of breast cancer might respond to chemotherapeutic or biological treatments for breast cancer. Most of the work that has been done in cancer research has used cells which have been grown on flat two-dimensional surfaces and hasn't really considered the three-dimensional aspect of breast cancer, which is a really important feature of cancers and the way that they behave. In our labs, we're growing cells in three dimensions in two ways. One is by growing spheroids, and the other is by taking cancer cells and growing them embedded in collagen matrix, compressing them, and then putting them into a more loose collagen matrix to make a sort of artificial cancer mass. We're then taking those three-dimensional cultures and using them in a flow environment in order to study the behavior of these cells in 3 dimensions and under flow, and look at the responsiveness of these cells to different chemotherapeutic and biological treatments. One of the things that's really important for us with our cancer research studies is having as close to a physiologically relevant model as we can possibly achieve. Clearly we're using the 3-dimensional cell culture. We're intending to add other cell types, which we've started to do by the way of fibroblasts and different cell types around the tumour mass. Alongside the flow system that we're using, it becomes a much more physiologically relevant system. This is really important in preclinical screening and drug discovery programs, and it will help to reduce unnecessary experiments in vivo and allows us scientists to really screen our drugs and our chemicals much more closely to the human system than had been possible previously. So as an academic in a department of Biomedical Sciences with colleagues who've got a range of diverse research interests, what's quite important to me when I purchase equipment is to make sure that it's going to be relevant to other colleagues who might be working in different areas. And for example, the Quasi Vivo system has been used by a colleague who's been interested in studying the formation of biofilms, another colleague who's been interested in looking at the blood-brain barrier, and other colleagues with different diverse interests that go alongside our own research, which is really focused on breast cancer metastasis and invasion of cancer cells. So this equipment is very useful in a range of different settings, and that's very important to me when I'm thinking about how to spend my money.